terápia következik, ami én azt gondolom, hogy rendkívül izgalmas terület lesz, úgyhogy nagyon sokan.
The next presentation is on the basics, the rudiments of gene therapy.
Hát köszönöm szépen a figyelmet, és
Thank you for the attention, and I would like to once again encourage parents and others, because encourage you to participate in these panels, because information collection is extremely important, especially in the sense that we have new therapies and we shouldn't only use these therapies. We also have to have feedback on them. We have to know if they help in the long run and what results they contribute in the long run. And for that, we need to follow these cases and get information. Now, gene therapy is one of the areas that has improved significantly over the past 10 years. As a neurologist, a neurologist typically liked to diagnose people, follow them, try to help as much as possible, but he or she couldn't really provide any therapies that would have actually helped improve the situation of the patients. However, the past 5 to 10 years has brought a lot of change, and neurologists have become much more optimistic. I think it's the best times to be a neurologist, because now we have new therapies that we can test and apply and use. And I think that basically, personally, I am just not thinking of retiring just yet, because this area is extremely exciting. Now, Albert Szent-Györgyi, the professor, Nobel laureate, has a quote: "Discovery is said to be an accident meeting the prepared mind." It's not about somebody being so brilliant that they just come up with something unique. It's more like interesting things happen, and then there are brains who are prepared enough to recognize those things as interesting. Now, Albert Szent-Györgyi lived very close to us in a biological center, and he was awarded a Nobel Prize for vitamin C, but actually he did more work in active myosin and muscle operation. That was something he was not rewarded for with the Nobel Prize, but his activities in that area were extremely important. Now, when I was very young, even in high school, I read James Watson's book, which was translated into Hungarian already, and that's when I decided I would not be an engineer anymore. It became an obsession for me, but Salvador Dali was also obsessed with the idea of DNA. He was completely in awe and tried to make various drawings of DNA. So when we talk about genetic abnormalities in neuromuscular diseases, we try to come up with ideas for the molecular basis of genetic engineering. And then I will also speak about DMD and SMA at the end of this presentation. Now, a central dogma of biology is that the information is stored in your DNA. It is transcribed into RNA, and then RNA information is translated into proteins. This happens in prokaryotes only. In viruses, this procedure goes on. I need to correct that. So reverse transcription can also happen in, for example, viruses, and that's something that we will be using later. Now, there are various mutations in the human body. Biology is not a perfect system. And this is actually the very foundation of evolution. There are spontaneous mutations. Some of these are advantageous, some of these are not. Sometimes your chromosomes are missing parts, some parts are duplicated, some parts are shuffled around, locations change. You see inversions. These usually realize as various syndromes or multi-gene diseases. Diseases are conditions that have been tied to several different genes. But also there are so-called silent mutations where you have a mutation, but it does not lead to any diseases. Now, there are some proteins which are generated in a non-incomplete form. There are mutations where the amino acid sequence is not what it should be. There are mutations that could be positive or negative. And of course, some of them lead to major problems in how the proteins function. So the result could be we lose functions, the functions become partial, so not complete. Sometimes you gain new functions which are, however, toxic. And sometimes there is a negative effect because you produce a protein that should be there, but there's too much of it and actually starts blocking some other functions. And there are RNA interference situations also when you have difficulties because gene sequencing techniques may not be able to identify these. So let's talk about the therapeutical situation, possibilities. We can change the activity of the DNA by what we call methylation, DNA methylation. We have drugs that cause non-specific changes in how the DNA is transcribed into RNA, which leads to a change in how the proteins are built. There are also small molecules that are selected from various cell cultures, or they could be small molecules that are created artificially. They can also be molecules that we find in nature that actually change how RNA is translated into proteins. And then there are artificial molecules, oligonucleotides, that change these functions. And also we have gene replacement. This is exactly what we call gene therapy. Sometimes you have an abnormal, non-functioning gene that you try to fix by introducing a copy of it into the system from the outside. And then you have also gene splitting or gene splicing, gene editing, where you can actually try and correct abnormal sections of a gene. Let me just give you one example for histone deacetyltransferase inhibition technique that we think will be is quite hopeful. We are part of a trial. Part of this goes on in Europe. And the Ital Pharmaco has a molecule called givinostat. We have had 4 patients that have been treated with this drug for 5 years now, and I can say that they're 14 to 15 years old, and they keep walking. which is an indication that givinostat is something we can use regardless what the actual genetic background is. The histology is very good, the muscle biopsy results are very good. So I think it might be actually approved as a medication, as a drug, hopefully, and it is something you can use for patients who are not the right type for other available treatments. Now, there are also point mutations that we have to somehow treat. There are what we call nonsense mutations, and we have a small molecule called atrophy. And we have been testing other drugs, for example, in the cysteine. Cystic fibrosis, and some of these molecules are approved in the US but not in Europe. But we have had a panel of 360 patients with Duchenne, and the jury is still out on whether this drug will be approved for Duchenne. Now, the clinical trials have shown that the motor function decreases, but it doesn't decrease as quickly as before. Almost, the effect is almost as good as with steroids, but it doesn't stop the deterioration. And FDA so far has been of the opinion that the impact is not big enough for the approval of the drug. But I think there is a big enough pressure in the US at least that it will be eventually approved. I have no personal experience with this drug, so we need more, but it could be one of the drugs that will influence or can influence the gene. Now, when we talk about antisense oligonucleotides, AS, that's a big research field. And let me just link this back to the AS antisense oligonucleotides, how these come into the picture. Now, these are short single-stranded DNA or RNA molecules. And Paul Zelensky was the first to apply these molecules. He tried to treat viral infections, and he realized that you can use a complementary section of DNA or RNA to influence natural processes. And he was retired from a research institute at 65, and then his biggest discovery came after he retired. So even if a retired person, even retired people can do a good job and create something very new. Now, ASO molecules can help in natural processes in various ways. For example, when we talk about the metabolism of RNA, this can be influenced by these molecules. You can block splicing factors. Splicing is when there is a split in the RNA during its maturation. You can either block this or you can improve this. So you can possibly use this in SMA and also in AMD in actually two opposite directions. Now, of course, you have to get these molecules into the cell, which is a problem. For the time being, we don't really know what the mechanism is. We don't know if there are any specific receptors or not. But what we know is that most of it that gets into the cell is actually eaten up by the it's just consumed and gotten rid of. But whatever gets into the nucleus may be built into the system. However, much of these molecules are, when they are introduced into the system, they are just basically destroyed already in the blood, and they never make their way into the brain, for example. So we have problems with that. Now, gene therapy is really based on the idea that there are basic strategies that viruses use. What viruses do is they have their own DNA and or RNA, which they do not replicate themselves. They introduce those DNA or RNA into the cells of animals or humans, which will either integrate into the DNA or RNA of the animal or the human. This is the typical case for RNA viruses. Or in the case of DNA viruses, it enters the nucleus, does not integrate into the gene, the chromosomes of the host, but stays there. Now, we were watching these tricks that viruses have been using for millions of years, and we have learned from them. So we created what we call viral vectors. Lentiviruses are typically used for this. They can introduce external DNA in diseases related to the liver and the bone marrow and blood production systems in the human body, where the cells continuously rejuvenate or are replaced, because you can only achieve a therapeutic positive outcome if you're just there all the time, regenerating healthy cells. Now, there is a difference in what situations, which types of viruses you have to use. Herpes viruses, adenoviruses, and the most approved, most accepted are the adeno-associated viruses. Those are most widely accepted. Some of them are able to transfer fairly long sections of DNA. Now, adeno-associated viruses are a very small type of parvovirus that do not cause diseases in humans. There are other parvoviruses that do cause problems. Now, these viruses have only 2 genes, and that's one of the problems. And there is a very short section of DNA that you can actually insert into this virus. So if you replace this section with the gene that you want to introduce into the human body, and then you can complement this in a cell culture, so you can grow viruses that have the type of DNA in them that you want to put into the human system, then you can just culture them and then get them into the body. This is the basic idea. Now, for gene therapy, you have to introduce these viruses into the human system. Thus, the new genetic information has to enter the cell in large quantities to make sure that this affects as many cells in the tissue as possible. And of course, the whole therapy has to be safe in terms of its procedure. Now, there are various ways to inject the viruses. You can inject them directly into the tissue. We have an approved gene therapy for eye diseases, which is a direct injection into the eye, which sounds a stretch, but it is happening. It's an approved therapy.
Témába, vagy pedig intratekális bejuttatásra tudunk.
There are other ways to introduce them into the system and so on, into the muscles, for example. But you also want to achieve the highest possible distribution to achieve the greatest impact. So intravascular and intrathecal, number 3 and number 4, are the best in the case of neuropathies. Now, just a comment on gene editing. The reason why you can change a gene sequence in vivo in the actual living creature is that there are specific bacterial nuclease molecules that can crop abnormal gene sections out of the gene, and then the body's own natural mechanism can repair that section of the gene. Now, the problem with this is that two different enzymes have been used for this, right here on the slide. These are sequence-specific, but you still have to help these enter the system. And these are bacterial proteins, so if you just express them within the system, within the body, they will be killed off by an immune reaction. So in order to be able to use this, you have to apply massive immune suppression, which is possible theoretically, but it is a major limitation. Nonetheless, it is important to create both animal models and in vitro models to find as much information as possible about how these techniques could be refined. Now, to be able to come up with a therapeutic or drug, you have to do large-scale testing in vitro as well as in animal models. You have to produce these drugs and these reagents in large quantities. You have to purify them to high specifications, and you have to make sure that these are safe. It is just a complicated, huge process. Not even the academic world is able to do this, so this is usually done by biotechnological companies in the market. And just a reflection on the history of gene therapy, and I'm not talking about the approved gene therapy procedures. A couple of protocols have been approved based on US data back in 1989 for a specific disease which used an adenovirus. Now, until 2010, most of these therapies used adenoviruses. There was a tragic event in 1999 which involved an adenovirus. There was a treatment that was done too early for a single patient who had a genetic liver condition, a young man of 21 years of age. And he was given a large quantity of adenoviruses, and he died within 2 days because of a massive reaction. Now, that death was something that was affected by in Montreal, because all the research was stopped for at least for about 10 years just because of that one death. So currently, the adenoviruses have been mostly just forgotten, except in vaccines, because they're being used in vaccines recently. But what is most commonly accepted today are adeno-associated viruses. Right now, there are major research studies impacting the central nervous system, as well as diseases of a neuromuscular type. We're talking about myotubular myopathy, limb-girdle muscular dystrophy, type 2 and type 3. The clinical trials are starting, and also in Duchenne's muscular dystrophy. But the only approved therapy exists right now for spinal muscular dystrophy. Now, let's talk about Duchenne quickly. You saw the protein here, and you know what it does. It stabilizes the muscle membrane, and most mutations are deletions and duplications of sections of the gene. Now, the question is, what is the difference between the Becker case and the more severe version of the disease? Well, there is a law that relates to the speed of transcription. In Duchenne's dystrophy, this rate of transcription goes down to 0, basically. In the case of Becker's syndrome, it continues, and there is a partial protein built, but not a complete protein. So one of the diseases is a missing protein. The other disease is a lower quantity of proteins produced, or incomplete proteins being produced. Now, this is the exon map of the DMD gene. This is one of the largest genes. It's 2 megabases, 79 exons. It's a huge gene, and the exons, if you take a look at the terminations, that's what influences the rate of transcription, whether it is in-frame transcription or out-of-frame transcription. Now, if just for you to understand it in more depth, if number 51, exon number 51 is blocked, then 39 and 51 will be out of frame, which means the RNA cannot continue the transcription process. It will just stop, like when you have a missing byte in your computer file. So how can we influence the situation? And this is where exon skipping comes in as a technique. If number 52 is missing, then you can block 51, and you can actually play around a little bit, and you can allow the transcription process to continue. You can test this in terms of checking the various mutations, and a good number of these mutations are candidates for exon skipping techniques. You can see a chart of them. The most commonly, exon number 51 is affected in 18%, 13%, 53 is 8%, and so on. And we are doing tests on the less rare, less common exons as well. Unfortunately, there is an expectation that whenever you do clinical trials, you have to focus on the very rare situations as well. It would be better, it would make more sense to at least introduce the therapy for the largest number of patients, and then we'll work on the rest of the patients later on. It doesn't always work that way. There are also a large number that cannot be of conditions that cannot be treated with exon skipping technologies. So right now we can only treat about less than 25% of the patients using exon skipping. Now, I'm sure one of the next presentations will speak about the clinical trial outcomes. We were part of a study that showed that motor functions are reduced over time, but this could be improved but not stopped, unfortunately. So the disease could be improved, but it doesn't stop. And this technique was only 5% better than exon skipping. It doesn't sound like too much, but it still helps improve motor functions. And new forms of exon skipping, which is called PPMO, is a lot more stable version of the same idea. It is more robust and a lot more effective. We are part of that clinical trial. We have a couple of patients who are being monitored, and instead of infusions every week, hopefully they will get away with just infusions once a month, and hopefully it will be more efficient than previous exon skipping approaches. And hopefully, if this technique proves useful or efficient, then the same approach can be used for other techniques as well. Now, gene replacement therapy means that you take a new type of gene section and you put that into the system, into the body. In the case of Duchenne, you have to rebuild the muscle, but not only the skeletal muscle, but also the heart muscle. So for gene therapy to be really efficient, you also have to treat the heart. Now, just talking briefly about my own work, I've been lucky enough to work back in 1989 when I first went to the USA, to Wisconsin. I started working in a laboratory where people focused on gene therapy. And just very randomly and accidentally, we discovered that when you put plasma DNA into the muscle by injection, some muscles will actually pick the DNA up and start producing protein based on those DNAs. Now, based on that, we used an animal model to express a protein that is involved in Duchenne. And this was basically a proof of concept because we showed that it is possible to make the DNA of a cell change and start producing proteins and put them into a location where you need them. But of course, the downside was that injecting DNA into the system is not efficient enough. But of course, we solved that problem when I started working with adenoviral gene therapy in Montreal, which was way more efficient, not only in muscle, but also in motor neurons. And then the problem was, like I said, that even though we achieved excellent dystrophin expression, and I had my wife as my co-worker, the problem was that there was a large rate of muscle inflammation as a side effect. So that moment on, we didn't improve very much until adeno-associated viruses came into the picture. And then, of course, you have various serotypes, and type 2 was way less efficient as type 6 or type 9, which is being used today, or the RH serotype, which is completely different. It is the rhesus monkey serotype. We learned that if you inject this virus into the vascular system, IV or into a vein or into an artery, then you can actually achieve a high rate of transcription in the muscle. That was the basis of our current muscular gene therapy. The problem in Duchenne is that the gene is way too long. It has to be split into a shorter form. And that means that we have a dystrophin in this vector that probably is involved in the Becker variety of this disease. So this will not be complete treatment. It helps, but it basically replaces a severe disease with a less severe disease. So if the system is successful, then instead of achieving a complete healing, we can decrease the severity of the disease by replacing it with a less severe, different type of the disease. There is a lot of work going on in nanomolecule research, and looks like there are better formulas coming up that will probably revolutionize virus-based gene editing. You have not here some efficiency data on that screen for this system. And right now we have a phase 3 trial going on for a specific adeno-associated virus technique. Most patients have already been recruited. And we see high rates of expression in patients. These are histological data for those involved patients, and you see high rates of expression. Now, this is a trial by Selepta, and I am part of the safety committee on this trial. We monitor what the potential side effects of this treatment are. Unfortunately, it is not without its side effects, but you can help them typically by immune suppression. There's one problem, though. We have learned that the patients whose mutation was at the beginning section, at the initial section of the gene within the first 17 exons, those patients will show a very high immune reaction when they encounter this shortened molecule. So for the time being, those patients are actually excluded. Nobody has qualified for the matrix of patients involved, for the pool of patients involved that have that modification in the first 17 exons. Now, some of these tests have only been done in mice, gene therapy using CRISP, and I think that's an area that we will be hearing a lot about in the future. Now, let me just very quickly move on to spinal muscle dystrophy. I don't really have too much time. I don't want to speak about the disease itself in a very lengthy way. There are different types of muscle atrophy, type 1, type 2, type 3, type 4. These are my own patients. Type 1 is the most severe. And when the outcome was tested, unfortunately, type 1 usually, if they are not artificially put on if they don't get oxygen artificially, then 90% of them would die by age 2. Now, this gene is on chromosome number 15. And what is interesting is that the very cause of the disease is the fact that SMN1 is missing recessively. Both copies actually are missing. And it's interesting that they have several duplication sections, sometimes once, sometimes twice, sometimes 7 or 8 times. And the more copies you have of these identical sections, the less severe the disease is. Interestingly, we do not have SMN2 in animals. We do not have a natural animal model for this, simply because animals do not have an SMN2 virus, gene. If an animal, the SMN1 virus gene is missing, then the embryo will simply die and not even be born. So how come humans can live without this SMN1? Well, that's because SMN2 allows us to replace some of the missing functions. It's interesting that human cells across our entire system are less sensitive to SMN1 problems, but motor neuron cells are more sensitive than others. So that means the central nervous system is not that heavily impacted, and that's the reason why this kind of genetic problem is mostly related to sensory and motor problems and diseases. We don't really have the reason, we don't know why that happens in humans. Now, SMN1 has a lot of functions, especially in RNA metabolism. Why we think the motor neuron is damaged is that there is an axonal transport here, which is a specific mRNA transport. The SMN protein is involved, and the longer the nerve and the axon, the greater the possibility of damage is seen. So as the axon itself grows, if there is not enough protein to support it, then its connection is joined with the muscle will not be normal. Okay, so let me show you SMN in a healthy individual. You have the normal protein being selected, but when SMN1 is blocked, then SMN2 will produce less of the proteins, or a less complete form of the protein, which blocks, which basically means that the embryo will not die in utero. And the fundamental cause is just a difference between a couple of base pairs. Certain researchers have done a lot of work in this area, and what we see is that in SMN2 there is a new splicing that takes place which blocks exon 7, and because of that there is a truncated protein being produced. Now, in addition to that, some of the protein will be healthy and the right size. So it causes problems, but less severe problems. Okay, so what we know about biology has told us how we can create therapies. We have to try to convince SMN2 to actually generate fully functional protein. And I think the way we can do that is get this splicing section and block it, so the complete protein chain is produced. This is the basis, the philosophy behind the technique before we actually, in combination with the technique specialized for introducing this DNA section into the cell. Now, Adrian Krainer is a Long Island-based researcher who was also taught by James Watson, who we have a lot to thank for, because he was the first to generate a technique to block that section of the process. And then later on, others contributed to all sorts of modifications, which led to a final therapeutic solution, the actual molecule that was sold to Biogen for commercial utilization. Now, this was tested in monkeys. If this oligonucleotide is injected into the veins, it will not enter the brain. But if you introduce it intrathecally, then it will be distributed fairly well. It will enter not only the spinal range, but also the entire brain. And it's also very good that this is a highly stable procedure. The intrathecal injection creates an effect, a stable effect for several months, which is very different from what we see in blood. So if you just use a few injections, it's enough to achieve a good therapeutic effect. Now, several trials have been done, and we were involved in EMBRACE type 1, type 2 for splicing modification using the molecule called nusinersen. But this is a chart of the summary data, comprehensive data for all the trials that are going on. And what we see is that the earlier we apply the therapy to an infant, the better the rate of efficiency will be. But this depends on how many copies of the SMN2 a patient has. And the people who had at least 3 copies reacted very well. People with 2 copies reacted well, but we cannot call that exactly healing, or exactly the outcome was not major for patients in type 2 and type 3. There is also a therapeutic impact, but not as good as when we introduce this technique after blocking the segments that cause problems. So once again, therapy should be applied to infants at an early age. And maybe I just want to add that in the case of type 3, there was a problem. What happens if somebody has this problem, gets this medication at age 17, 18, 19? Now, based on 2 patients I have, I can say that the therapeutic impact is dramatic. So this medication will probably stabilize the motor neurons that are still operational but are down the slope already. Which means even if you can apply the drug later on, you should, because it still helps. Now, coming back to gene therapies, in animal trials, it has been very effective. And in human gene therapy, we can at least continue working on it. Similarly to nusinersen, the earliest we use gene therapy, the best. We have had trials with patients under 2 years of age and some above 2, but this is very difficult from recruitment perspectives because we can only use patients who have not been treated any other way, and that's not easy to find. And I would like to finish my presentation with that little footage right here. Thank you.
Nagyon szépen köszönöm.
Thank you so much. This is a highly complex issue, and I think we would like to listen to you for longer. But I think we need to have a little break now. So if you have any questions, please remember them and ask them later on. And I'm asking the technicians to prepare Laurent Servet, professor's pre-recorded presentation, who is a fellow with more than one university, and he is going to shed light on the pharmacology aspect of the issue. And I can see people standing up. Yes, this has been a long morning session.